I thought it's very important that we have a good performance evaluation of equipment, which is an integral part of quality control in any laboratory. And therefore, I'm going to be speaking on equipment performance evaluation. And unless this is so foolproof, we cannot rely on it. So what are the various aspects related to equipment performance evaluation is what we will be discussing. So primarily the discussion will focus on an overview of the evaluation of hematology analyzer with various aspects of quality control procedures, discussing certain issues concerning standardization of clinically important CBC parameters and the need for reference method as well as harmonization. So an approach to quality control requires that every factor in the instrument performance be appropriately controlled. The procedures that should be adapted are based on the use of control materials, correlation assessments, and statistical analysis that we routinely perform. So the role of an hematology analyzer is not just to speed up the workflow in a laboratory. However, it improves precision as many cells are counted. The cell classifications are based on measured objective properties of cells. Leukocyte differential counts are much more reliable, and therefore it provides clinician with more reliable data for proper management of patients. So the parameters that are available to us is a huge battery which includes nucleated red cells, immature granulocytes, different cell indices, red cell parameters, platelet parameters, and in today's diagnostic arena, digital imaging analysis as well. So, in fully automated equipments, control is required of the equipment to ensure that it measures up to the manufacturer's specifications and that it's correctly calibrated. There are, however, smaller labs which use semi-automated equipments where, assurance of technical skill of the operator, the reliability of the reagent and the test performance are equally important. Diagnostic hematology is based on morphology, making stained blood films. Not all laboratories are as advanced as CMC, and therefore we still rely on morphology. Therefore, the role of peripheral blood smear still plays an important role, and their interpretation requires professional judgment. So how to choose an analyzer becomes an important decision. Of course, we do rely on the instrument manufacturers to provide us a lot of information. However, peer-reviewed reports on comparative evaluations play a significant role in us deciding the choice of the analyzer. There are several other factors that contribute to this decision-making: the dimension of the equipment, information related to consumables, the ability of the instrument to be interfaced with the hospital laboratory information system. All these factors contribute to this decision-making. And in addition to the repertoire of parameters that are available, the minimum sample volume, the data presentation, etc. So the new instrument should generate results that are comparable both in the units reported as well as reference ranges that are already in place in the laboratory. Of course, it must be evaluated with both normal as well as pathological samples before introducing it into routine lab practice. So that's when the term validation comes into picture. So when we talk about validation, it's establishing documented evidence that provides a high degree of assurance that a specific process will consistently produce results meeting its predetermined specifications and quality attributes. The data obtained in one laboratory on one instrument should give comparable results to that provided in another laboratory. This is very important. So the ISO 15189 states that the laboratory shall verify upon installation and before use that the equipment is capable of achieving the necessary performance and that it complies with the requirements relevant to any examination concerned. So the verification process should be performed. It should include precision, accuracy, analytical sensitivity, which means limit of detection, analytical specificity, including interfering substances, reference ranges, patients' correlation studies, limit of detection, carryover. These are the important aspects that we verify before the equipment is put into routine use. These objectives shall be investigated using well-defined protocols and their performance validated against acceptable reference method. And in the absence of these reference methodology, an on-market analyzer is used for comparison. So verification is the confirmation of evaluation performed already by the manufacturer. Verification should provide evidence that the analyzer can meet specific requirements at the test site. The performance attributes of the equipment should be obtained in the environment where it's going to be sited and all the staff who will be operating it. The reference ranges must be established by each laboratory before the analyzer is used for patient testing. So we know that the guidelines state that the responsibility of the instrument installation, setup, initial calibration using specified methodology essentially lies with the manufacturer. However, once this is achieved, the end user shall assess whether the manufacturer's claim on performance of the specific instrument also apply to the intended use criteria by the laboratory. And this process is called verification. So the parameters should be verified by the same procedure as that used for validation. We routinely use anonymous surplus samples that are available in our laboratories to determine the degree of precision and at medical decision values. That's very important because these can play a role about transfusion threshold decisions. So samples should be selected to ensure that extensive variation of pathological sample is included in the analysis, and samples in these situations sometimes you could even dilute these samples to acquire a broad range of results. So when adapting a new hematology analyzer, it's important to compare this with the existing equipment to ensure that the patient's results are not affected. So the scope of verification depends on the parameters reported by the laboratory and the range of samples that are available to us. Of course, there are challenges unique to hematology, difficulty in establishing reference preparations owing to the relative instability of various hematological parameters. So when we talk about quality control procedures, in addition to instrument calibration, routine check with control samples, use of patient sample, preserved controls, stabilized blood cells that are available to us, provided by the manufacturer, and statistical analysis, correlation assessment contribute to the QC procedures. So when we talk about the cell counting control, what are the characteristics that we look for? Of course, it should be stable, easily suspended, does not agglutinate. It should be, the rheological constants should be similar to the blood that is routinely used. The controlled particle size and shape contribute to these characteristics with specific conductivity of the particles and the medium. So calibrators are essentially used which have stated values and are used primarily for equipment calibration, whereas controls are used for replicate testing within a laboratory on a day-to-day basis. So when to calibrate is another question, of course at installation and year after year, and as recommended by the manufacturer. There could be situations where we have major equipment breakdowns or when quality control data show there is something seriously going wrong, and there could be a complete change of reagents. In these situations, it's important to recalibrate the equipment. The instrument manufacturer provides installation, calibration, and performance validation at the time of installation, and this is verified by the end user as a laboratory, and as I already mentioned, normal and abnormal samples to be included. The records of performance verification are to be maintained appropriately. Original printouts are necessary to be kept as reference, including record of downtime, breakdown, service response times, maintenance schedules, operator log. All of these are essential. Safety considerations do play an important role. It's also important to keep in mind that sample modes, when we have more than one mode, most of the equipments do have automatic and the manual mode. It's important to verify the performance of both the modes for all the parameters that we already spoke about. The reliability of barcode readers should also be monitored throughout the evaluation. So when we refer to precision, It is the closeness of agreement between test results when a sample is run repeatedly. It's obviously not a measure of accuracy. Imprecision increases in samples with lower cell concentrations as fewer cells are counted. It can be reported as SD and CV percentage. So an increased SD and CV percentage indicates an highly imprecise sample. Precision should be established for the entire reportable range of a parameter, and this would obviously include low counts, especially for hemoglobin and platelet, as I mentioned, at the transfusion threshold. Within run precision is also equally important. It's important to include normal and abnormal samples that we referred to. Linearity is another aspect that we have to perform. Ability to provide results that are directly proportional to the concentration of cells is what determines the linearity. So the question quite often that comes up is, what should be the frequency of linearity testing? So this is an example of linearity testing for various parameters. Sample stability is another important issue. Ability of a sample to retain a consistent value for a measured parameter over a defined period of time and within specific limits when stored under defined conditions is sample stability. So anticoagulated fresh whole blood should be processed within 4 to 6 hours after sampling if stored at room temperature. However, if samples are refrigerated, they are generally stable up to 24 hours, after which evidence of deterioration sets in. If you use pediatric sample or capillary sampling, there are other factors that need to be kept into consideration, especially if there is insufficient mixing or problems related to dead volume of the hematology analyzer, then the results may not be very accurate. So the closeness of agreement between results of a measurement and a known true value is what is accuracy. All the hematology parameters need to be measured for accuracy. So the reference method becomes important. For hemoglobin, of course, we do have a reference method, that the hemoglobin cyanide method, flow cytometric reference method for platelets as well as reticulocyte. Differential count, we still rely on morphological verification by two morphologists, count of 400 cell count, and that is still, I think it's still the reference method. However, we do understand that there are limitations. The slide distribution error, the interpretation error could contribute to this variation, and I'm sure there are better methods that we could rely on. So when we talk about comparability, regression analysis, correlation blind elements plots are used to assess the extent of agreement. Correlation coefficient is a measure of how well the data falls in a straight line. Closer the correlation, more linear is the agreement. The slope, the intercept, and the bias assessments are all additional useful information that we look to. So, as was heard in the previous talk, it's important to look at the sensitivity and specificity of the flagging. This is determined by comparing whether the flagged or unflagged samples accurately distinguish samples with and without morphological abnormalities, based of course on morphological review. So the accuracy of flagging is determined for various clinically significant parameters. Sensitivity is important to identify all abnormalities, and therefore we produce accurate results. However, specificity is important to limit unnecessary microscopic examination and thereby increase the efficiency of the laboratory. So it's very important to have the striking delicate balance between the sensitivity and the specificity. So instrument flags play an important role. So you could have manufacturer control flags which are already available to us, laboratory control flags for critical decision making rules and critical ranges is incorporated by the concerned laboratories. There are several messages that are provided to us. For example, abnormal cells, red cell agglutination, platelet aggregation, and so on. So laboratories using these analyses should create policies for slide review and manual differential count. Carryover is another important aspect that we should keep in mind, because this can erroneously result in a falsely high value in a subsequent test sample and leading to erroneous clinical interpretation. So the limit of blank refers to any signal that is measured that actually does not originate from that particular sample and a parameter of interest. optimally, the background counts have to be 0. This is just an example to show that we quite often, if there is no proper background testing, you can have erroneous platelet counts. The next aspect that we need to discuss is the calculation of reference interval. So, as desired by the standards, it's important that each laboratory establish their own reference intervals. However, having said that, it's going to be a challenge, particularly in pediatric patients. So, published reference intervals and those provided by the manufacturers are quite often used by all the laboratories. Inter-instrument comparison is another important aspect. So, if a laboratory has multiple analyzers and different analyzers using different methodologies, inter-instrument quality control is important. This is to ensure that we don't have unexpected discrepancies between results generated by these equipments, resulting in unnecessary clinical intervention. So this can be done by running patient samples across the analyzers in addition to the routine quality control testing that we perform. So for intra-instrument quality control, QC samples on a daily basis is necessary. These are obtained easily provided by the manufacturer. So these are run after calibration of the equipment and periodically on a regular basis during testing with appropriate storage conditions. That becomes important. The next question is, are there pitfalls with the QC materials that are available to us? The manufacturer's target limits are often very broad. Thereby, subtle changes in the analyzer behavior can be missed. Therefore, it's recommended to adjust the target range after the laboratory runs several runs and measures its own mean and the target range. A benefit in using these QC samples, as we all know, is we can identify drifts using the LJ graph. So the daily controls, commercial control, patient control, moving average, correlation, comparisons, and policy for acceptability of QC testing, troubleshooting, all are various aspects as an integral part of IQC. So therefore, we should have policies and procedures, assign staff responsibility, establish control ranges, monitor control values, and develop procedures for corrective action. So this is something well known, so I'm going to quickly skip this. Yeah, so the important question, as I was just talking about in the previous slide, can manufacturers' stated reference ranges be used by the laboratory? Yes, the standard requires, as I mentioned, that the laboratory measure its own reference range. However, there is an allowance to use manufacturer ranges when the following conditions are met. The stated values correspond to the method and instrument used by the laboratory. The mean obtained by the laboratory reflects the manufacturer's stated mean, and the laboratory assures that the range is narrow enough to detect clinically significant errors. So manufacturer ranges can also be implemented if a test is used sparingly, infrequently, and that calculation of valid statistics may not be possible. In a setting where there is high reproducibility, the laboratory's range can be very small. In those situations, this can result in unnecessary repeat testing of QC, and because it's outside the laboratory range, however, it is within the manufacturer reference range. Therefore, it's important that manufacturer selection of appropriate range is a very delicate balance between these two spectrum, that provided by the manufacturer and that generated by the laboratory themselves. So we should also note that certain parameters like MCV, the QC material that we use, the MCV gradually tends to increase with time, the red cell values decrease, platelets values increase. This is because the cell volume increases, the RBC starts lysing and get counted as platelets, resulting in erroneous results. So QC should be evaluated, of course, monthly to ensure continued acceptable performance. There should be a QC policy for trends, bias, and shifts, and establish all this when calibration is required. So when QC goes out of control, it's important to stop testing and identify the problem. Do not report patients' results. There has to be a policy for troubleshooting when results go out of range. Retained patient sample testing can contribute significantly to a part of QC procedure. However, please note that these are additional approaches to IQC and not alternate approach. So performing duplicate tests on patient samples can add value For example, if you can perform duplicate testing and determine the degree of variation, and none of the duplicate tests should differ from each other by more than 2 SD. This can be a very useful internal QC parameter. Moving average is another useful measure. Daily mean, of course, correlation comparison. Delta checks that we all rely on. It's important that delta checks could be crossed when there is pre-analytical issue or possibly clinical therapeutic interventions. However, delta checks, if monitored on a daily basis, play an important role. So automated reticulocyte, again 2 levels of control with at least verifying the manual performance once a week can add to our QC plan. So when we plan IQC, the size of the laboratory, number of specimens, whether the lab is a 24-hour lab, the type of equipment, accreditation requirements, all of these factors should be kept in mind. So these are the various QC tools that we just spoke about. So I would like to conclude the session by drawing your attention to certain important aspects. So CPC parameters that we use, not all of them have reference methods. So that can pose a challenge. MCV and other parameters, there is no reliable reference method. Platelet parameters, there are many pre-analytical variables that come into picture, so that can be challenging. There can be difference between 2 laboratories generating these results because different analyzers use different cutoffs and measurement methods contributing to the variation. So to summarize, the verification process of a hematology analyzer by an end user laboratory involves issues with regard to pre-analytical and analytical phase, which should be standardized and controlled. Reference methods and standardization of CBC parameters will contribute to harmonization and more reliability on these results generated by the auto analyzers for clinical practice. With this, I conclude the session. Thank you.
Thank you very much, Dr. Sita Lakshmi. Dr. Manglesh, over to you for any questions, Q&A.
Thank you, ma'am, for a lucid talk. I think any questions are not remaining, but still, if any doubts, please.
Yes.
Ma'am will happy to answer.
Yes, I have a question. One of the, let me say, problems or is how we can control the transport of the samples from laboratories that receive samples from outside. And of course, the temperature can change. It's not sometimes room temperature, and can be sometimes 8 hours, 6 hours, 10 hours. And my challenge is that how you propose to we control this transportation of the samples.
Yeah. I think this is a very important aspect that control becomes important. I did not touch too much on pre-analytical aspects. However, this question having come up, it starts with appropriate sample collection, how the samples are mixed, and what control is maintained during transport. So the temperature control, of course, especially in India as a country where you have regions with significantly high temperatures as well. So monitoring and transport is an important aspect. So temperature, how the samples are kept and how they are mixed after collection and appropriately transpose the volume, the ratio of anticoagulant blood, all this can contribute to the extent of variation that we will generate from the analyzer. So you have brought out a very important point, sir, the pre-analytical, especially when samples are received from different locations to a laboratory, can pose significant challenge because very many conditions we may not even be aware because we may not have even witnessed the site from where these samples are coming. So this can become, this can contribute to erroneous results that we produce by the analyzer. Yes, thank you.
Yes, as a proposal from me is that when you realize that you are going to receive samples from a center,
yes,
I recommend to emulate the transport, sending a sample collected in your own lab, sending to the half of the time that you need to arrive to this place and return to your lab and measure again. Because before to approve to receive samples from a center, I think some controls have to be done to evaluate really the changes and to realize if you have to transport it at 4 degrees with a special transportation boxes or not. And my second question is about the morphological parameters. For example, parameters that are not measuring directly, and it's difficult to have a control that is the same, for example, IRF, IPF, morphological parameters from the graph that you cannot find any analog to emulate exactly a reticulocyte to be analyzed. How we can control that, and this is also another question.
So in this context, what I feel is we don't go by the results generated of a single parameter. Like sir was talking about in the previous session as well, hematology is in the context. So we look at it in the context of the case, correlating with other parameters, which can add value to reporting rather than go by the result of an isolated parameter. I think I would place a lot of importance to this aspect before we generate a result. But however, this can be challenging in certain laboratories where results are just, samples are fed into the machine and results are generated without this aspect. Having said that, lack of adequate clinical information that is received in every laboratory can pose a challenge if we interpret these results in isolation. This is what I think.
Thank you.
Any other question?
Dr. Sita Lakshmi, very good compilation of a quality system of lab. We have a lot of pediatric samples and finger prick samples. Now to do the quality control of those, you run the controls as a whole blood sample. What is your thought on the finger prick sample of calibration of your equipment and the validity of those results? First question. Second is on the inter-instrument manufacturers' level of platelet counts on the basis of the size of the platelets. Now we have many communities who have higher giant platelets. When they come to one analyzer, it gives 30,000, the other gives 80,000. Both are low, but look at the variation. It is the discriminators that these manufacturers are putting in their equipments. Should there be a uniformity in that as well? Two questions.
Ma'am, I think you have just hit on the nail. In fact, that's what my concluding session was. There is an urgent need or absolute need to harmonize the results generated because there are analyzers, all of them are good in their own way. Each of them have different methodologies of testing. But however, in certain clinical situations, we can have discrepant results because of these variations, and there is lack of reference methodologies that are available contributing to the problem. So you said it very right, and I agree 100%. There is an urgent need to harmonize and standardize. Till such time, I would think the answer to both your questions, I would still rely on peripheral blood smear, morphological microscopic examination of the smear till we reach that stage. This is my response, ma'am. Thank you.
Thanks, ma'am.
Thank you.
For a nice morning academic feast.
Thank you, sir.
I think this will help, especially to the younger ones who are freshers in the field of hematology. Thank you, ma'am.